Hello, my name's Lindsay Turnbull and I'm an Associate Professor in the Department of Plant Sciences at the University of Oxford. And we're right in the middle of this very serious coronavirus crisis right now. And my students are all stuck at home and we want to keep them in touch with biology and keep them in touch with us. And so we're going to make a new series of videos and they're going to be called Back Garden Biology. Hello, welcome to this week's episode of Back Garden Biology. I'm down at the shed end where we had the garlic mustard and this time I'm looking at another plant that I've just allowed to grow. I haven't planted either of these plants here. The most obvious thing you can see here are Spanish bluebells, but I haven't planted those. They were just here in the garden when I arrived, as was this strange thing with the very large green sort of arrow-shaped leaves. With the thing at the bottom like that, so it's like a big arrow shape. This is Arum maculatum. It's a very common native British plant that grows in woodlands and can often be found in gardens. And again, normally treated as a weed, but here at the back of the garden, I like to leave things like that and just let them do their thing. Although I do dig it up when it comes up in my flower beds, I have to admit. Let's have a closer look at one of these plants. What's interesting about it now is it's flowering. So you can see that extraordinary looking structure appearing in the middle. That is the inflorescence. It's all closed up tight at the moment. And as it opens, you can see it change. So I'll just cut off an open one now and we'll have a look at it. So unfortunately, the only ones that are open are facing away from us. So I'm going to cut one off right at the base and show you what it looks like. And it's this extraordinary looking thing. So you can see that this part opens and in the middle there you have this extraordinary structure called a Spadix. I don't know if this is a Spadix or a Spadix actually. And it is said that this, on the day it opens, it's supposed to start producing a stench. Oh, I can still smell it on that one actually, and it's a stench, and it is a stench, not a delightful aroma. It is a stench of stale urine. That's how bad it is, and it's trying to attract pollinators, believe it or not, tiny little things called moth midges, and in there is where the business end of the flower is. So the inflorescence, the pollen and the female flowers that need to have pollen transferred onto them are tucked away inside here. And we're going to dissect one and have a better look. But what's interesting and what we're trying to do at the moment is on the day this opens and this smell starts to appear, the Spadix here is supposed to start heating up and you're supposed to be able to measure that. So we're out here with our thermometer trying to measure, does this heat up, can we measure it, and just how hot does it actually get? So what I'm going to try and do now is we've bought a little temperature measuring device. So it has a screen that displays the output and connected to it are two thermocouples which can make reasonably accurate measurements of the temperature. And we're going to see whether that Spadix inside the spade, the brown spiky bit, whether it really does heat up. Now we've actually been trying all through the day to measure this on one that we thought was a newly opened flower and not measuring anything very convincing. It's now 6 o'clock in the evening. We came down to have one last go because this is the time when they're really supposed to be heating up to the maximum. And we realised that a new one had just opened and the smell from that one was incredible. So that's the one we're going to try and measure now. We think actually the other one was a little bit too old. So let's focus on the screen. You can see you've got a top measurement there of 19.1 and a bottom one of 19.2, 19.3. So they're both giving a very similar measurement. And this is T2, so this is the lower one. So this should be the lower reading. I'm going to place it against this newly opened Spadix and see if we can see the temperature climb. So Let me have a look then. Oh my goodness, I was try hard to keep it in contact with the Spadix. So it is around 25 degrees when ambient temperature is only around 19. So it's managing about 5 degrees C above ambient. And I think that's pretty impressive. You know, this is a plant. It's not a mammal. It's very unusual to have a plant that can heat itself up like that. And it's no coincidence, of course, that the smell off there is now so powerful. That's why it's heating up, so it can disseminate that smell as widely as possible. So let's go inside and dissect one of those flowers and see exactly why it's bothering to go to all this effort. So I cut off this inflorescence. I'm going to have a go at dissecting it. I have to admit, I've never done it before, and I've got a little paper, scientific paper next to me, which describes what I should be finding inside here. But I'm going to see whether I can find that or not. So this is the spathe, and the brown thing is the Spadix. Now, I didn't say what this plant is called in English. I gave you its Latin name, Arum maculatum. It has a lot of common English names, some of them, frankly, far too rude for me to reveal in this film. But it's often called now either lords and ladies, which all sounds very clean, or cuckoo pint. And when I tell you that pint is the shortening of the old English word pintle, which means penis, then you can see why a lot of these names are, well, not fit for family consumption. But of course, medieval people were much more robust and bawdy than we are now. Okay, this whole thing is a trap. It's a trap for those moth midges or owl midges. They are attracted by the smell. They then go down into here where they are trapped for the night and forced to pick up pollen and move pollen around. And as the thing withers away, then they're released again. So I don't know what state this one's in. I don't think it's been open for very long, but it didn't just open today. Okay, let's open it up and see what we can see. So I'm going to peel this away, see if I don't have to cut it, but if I can just get it by peeling it. Got to unfold there, and I'm going to unfold here. Oh yes, and look what we can see inside here. That is rather marvellous. So you can see, I'll try and hold it as still as I can, at the bottom of the Spadix is a first ring of hairs. That is the trap. So, and here is, if you can see in there, there is one of these tiny little moth midges or owl midges crawling out and crawling up and away up the Spadix. So that is what the plant was trying to do, and it succeeded. So you've got this ring of hairs. These are the male flowers then, the little brown ones that produce the pollen. And this cluster at the bottom are the female flowers. And there is some nectar down there to keep our little owl midge alive. And there's a second sparse row of hairs there, which also helps to keep the insect in. So isn't this amazing? You know, I always think that we pay a lot of attention to you know, picture plants and exciting plants that you can find, and you've got to go a long way to find them. But this is something in your back garden, as clever a thing as you could ever hope to find in the Galapagos Islands or anywhere else. And what's even more amazing is that there is just a single species of moth midge that this plant depends on. Only one species basically pollinates this plant, and it just goes to show how fragile a lot of the connections are, the ecological connections out there. And that's why it's really important to preserve as much as we possibly can, because most things are doing something, even if we're not always clear exactly what it is. I want to think for a minute about how that arum produces that incredible heat. And when you see something like that, you know that there must be something going on in the cells that underpin it. So I want to think about a plant cell for a moment and how it works. So this tray on the table here is going to represent my plant cell. Plant cells are square, rectangular, because they have a rigid cell wall, which we're not going to focus on this time. And here are some of the main components of a cell. This is supposed to represent the nucleus, and inside there we have the genome. That's all the information that the cell needs to carry out all of its activities. But an instruction manual is no good if there's no one to carry out the instructions. So the cell also has what we can think of as key workers. The key workers are called ribosomes. We're hearing a lot about key workers at the moment, and inside a cell, the key workers are ribosomes. This is not to scale. They would be incredibly tiny. So their job is to translate messages. So from the genome, messages come out, and the ribosomes will clamp onto it. They look a bit like a little snowman with a small bit and a large bit, and the message goes in between. And they will then read the message and they will build the component that the cell needs. And all those components are made out of something called protein. And it's their job to build the proteins by sticking together individual building blocks called amino acids. Now that process, which we call protein synthesis, demands a lot of energy. So the cell needs to produce energy. And the way it does this is to provide the equivalent, it's a molecule, it's the equivalent of a battery. And that molecule is called ATP. And those little batteries can be plugged in here, zap, 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 to fuel this process. But once they've been used, they have to be recharged. And that goes on somewhere else in a cell. So in an animal cell, we'd have a lot of these things called mitochondria. And those are where the batteries are recharged. And the animal eats sugar, eats food, and the glucose is sent here. And that glucose has lots of energy stored in the bonds. And if you rip glucose apart, you can release that energy. And in the mitochondria, the energy that's released by destroying glucose is used to recharge the ATP batteries. Now, plant cells also have these mitochondria. Now, plant cells, of course, are famous for being green, and that's because, unlike animal cells, they have an extra structure in the cell, and that's called a chloroplast. And these harness the sun's energy and produce glucose. So they don't have to eat anything, they're making their own food, but they still have to recharge ATP batteries. Some of that can be done here by the chloroplast itself, but a lot of it is done here. And of course, in the dark, the cell has to keep running, has to keep making things, and it has to rely on these mitochondria. Now, this process of ripping apart glucose and recharging ATP batteries is called respiration, and all cells do that. Now, what's interesting in the arum is it's got to generate a huge amount of heat. And as we said, normally the mitochondria in a plant cell are not running at really full tilt. They don't need to recharge billions of ATP batteries all the time because the plant is not expending as much energy as an animal cell would. So these are quite just ticking over, if you like. What the arum wants, though, is to produce a huge amount of heat. And although that process of respiration will produce a small amount of heat, it has decided to do something different. So what it does when it sends the glucose to the mitochondria is it's got a different pathway that it can send them down. So instead of the energy being harnessed to recharge ATP batteries to do normal cell stuff, all that energy is just wasted as heat. So they've uncoupled the process of destroying the glucose and normally that energy being harnessed to something productive to just letting it all be generated as heat. And for a couple of hours only, that plant cell is blasting through glucose at a similar rate to the muscles in the wings of a hummingbird. So just for a couple of hours in its life, once a year, that arum really takes flight. So we're just finishing this video where we began at this little patch of Arum maculatum. You can see this one here compared to even 24 hours ago. The inflorescence is swelling up a bit more, and I suspect at 6 o'clock this evening that is going to open and the smell is going to start and the heating up process is going to begin. So I really hope that you get a chance to have a look at this. I mean, it's really one of the most amazing things I've seen. I know about it, or knew about it, but I'd never actually gone out and had a really close look myself. I'd never tried to measure the temperature myself. And it just goes to show how all of us, even professional biologists, have become more and more disconnected from the real organisms, and we spend far too much time indoors. So good luck with your arum lilies, and I hope, you can get some temperature measurements yourself. If you don't have an accurate thermometer, apparently you can even, just by holding the Spadix, you can feel the heat from them, and 6 o'clock in the evening, that's the time to go and do it, and try to find a newly opened Spadix. You'll know it is because of the smell coming from it. So good luck with that.